Today we will be discussing Mattiroli's investigation of the structure of histone-based chromatin in the domain of Archaea. This is a phylogenetic tree representing the 3 domains. This study focuses primarily on Archaea and its similarities to Eukarya. In eukaryotic cells, DNA is wrapped around histones, the group of basic proteins found in chromatin. Histones enable DNA folding and compact the genetic information, facilitating more practical storage. Nucleosomes are formed around a core of 8 of these histones, a structure known as a histone octamer. It is important to note that eukaryotic nucleosomes are limited to just 8 histones. DNA is negatively charged while histones are positively charged. This results in the electrostatic attraction which holds them together, as designated by Coulomb's law. Eukarya packs the DNA histone complex in the following process. Chromatin begins as a double-stranded superhelix of DNA which wraps around a nucleosome of 8 histones. The nucleosome with the DNA joins with an additional histone to form a chromatosome. These structures then form strands and loops which are compacted, folded, and closely packed to form the chromatid of a chromosome. Histone DNA complexes in Archaea are not limited to just one structure in the way that Eukarya are limited by defined nucleosomes. Archaea can spiral around several stacks of histones and form extended structures, creating super long helices as a result. The purpose of their experiment was to figure out if the methods of DNA compaction in Archaea were similar to Eukarya. To achieve this, the researchers disrupted the superhelix structure of Archaea in vivo while preserving the DNA binding of histones.
Experiment. X-ray crystallography reveals the locations of nucleotides and amino acids in order to find where the disruption of the DNA and dimers was needed. Researchers used the DNA sequences and homodimers of Methanothermus fervidus and Thermococcus catacarensis, both of which are species of thermophilic Archaea. Our representation here is modeled after Figure 3b from Mattiroli's study. TS600 is the strain of Thermococcus catacarensis that has an intact DNA histone complex, while TS621 is the strain that has been disrupted. This demonstrates that TS621 has decreased frequencies of normalized fold expression, which signifies the importance of the DNA histone structure in terms of function. This representation is modeled after Figure 3c in Mattiroli's study. This shows the reductive effect on fragment sizes measured in base pairs as a result of disruption to the histone DNA complex, further exhibiting the importance of this specific structure in terms of overall genetic function and spatial organization. The results of this experiment lend credence and evidentiary support to evolutionary theory by demonstrating that the means of DNA packaging in Archaea and Eukarya are very similar. Figures 3b and 3c use the data collection methods of normalized fold expression and fragment sizes measured in base pairs. Both figures demonstrate how the DNA packaging mechanism of Archaea is negatively impacted by a disruption to the connected histone complex. Irregular folding and smaller fragment sizes in base pairs are the result of this disruption. This is representative of the idea that structure is vital to function. The DNA histone structure is necessary to the function of DNA replication and protein folding that pertains to it.
Mattiroli's study demonstrated that Archaea and Eukarya share a common ancestor in regard to the storage and packaging method. This experiment emphasizes the significance of polymerization of amino acids into polypeptides and how peptide bonds are related to DNA packaging.
The histone-based mechanism of DNA compaction predates the nucleosome of Eukarya. There is much to be learned from microbiological processes in terms of how holistic conclusions regarding taxonomy and evolution can be drawn.
The relationships between the modules concerning evolution, ecology, and cells are predicated upon their ability to coalesce and explain each other's phenomena.
Biology on a macro level cannot be explained coherently without the evidence of the micro level and vice versa. In order to support theories in both areas, one has to build a solid foundation. Gathering more information about fundamental biochemical processes like the ones in this study help tie together these subfields of biology.
Studies in microbiology like Mattiroli's help to explain how evolutionary processes occur and why species have the traits or interactions with other species that they do. It has helped to answer certain questions in biology while also generating new questions with respect to evolutionary theory.
Future experiments may strive to answer questions about the processes by which this more primitive form of chromatin structure seen in the study evolved into what we see in eukaryotes. Thanks to Mattiroli's study, we have gained insight into the similarity of Archaea and Eukarya in terms of the DNA histone complex. Consequently, this expands our knowledge about the evolutionary relationships between these 2 domains and their common ancestor.
Ultimately, this leads us closer to answering one of the most fundamental problems of modern biology. What was the last common ancestor? The advancement of knowledge and theory resulting from studies like these better connect us answer the questions that biology is all about.